think what probably makes the most sense in terms of segueing, since we've just been all talking about urinary system, is let's talk about the pre-lab for Tuesday, just so you have a sense of how to approach that. And then we'll dig into the spirometry. So if you go here on in the module, it says, kidney regulation of osmolarity lab pre-lab. So this is going to be the one we're doing next Tuesday. And the basic idea here is we're going to have four groups of people. And as an independent variable, we're going to change what they're consuming, what they're putting into their body. Some people will have more or less water. This 800 mils is like a big gulp. It's like a large cup. 80 mils is a tiny little Dixie cup. It's not much at all. Sodium chloride consumed. Some people are going to be taking salt tablets and some people won't. So that's another independent variable is, ingestion of salt. Another one, ingestion of caffeine. We're going to have one group have caffeine and other ones not have caffeine. So does everybody have a basic idea? We have these three independent variables, the ingestion of either water or salt or caffeine. And then what we're going to do for the people in these different groups is monitor the volume and composition of their urine. We're basically we're going to be collecting urine every 30 minutes. So after they take their treatment, whatever they are supposed to do for their group, then every half hour they have to pee. And even if they don't, they have to try to pee. Even if you don't think you have to pee, you can still pee a little bit. Like I said, the bladder doesn't initiate that stretch reflex until it's got about 200 mils of urine inside. But so basically every half hour, we'll see what is the volume of urine in this collection. We can look at the specific gravity of the urine during that collection. Instead of using the multi-sticks like you saw on Tuesday, we usually in the lab, we use that little hydrometer, that little glass float. So you'll get to see you'll see a video of how that works later. But for your hypotheses, you don't have to worry about how we're getting it. You just have to know what it is, density. And then we're also going to test for sodium chloride concentration. So these are our three dependent variables, right? The volume of the urine, the specific gravity of the urine, and the salt concentration of the urine. So does that make sense? The basic setup is these independent variables, the ingestion of water, salt, and caffeine. And the dependent variables are the volume and composition of the urine that's being formed. And so what I want is a hypothesis for each of the four groups. So for group one, what do you expect in terms of these variables, volume, specific gravity, and salt? For group two, what do you expect? For group three, for group four? Notice we're not going to be able to do quantitative stuff. It's going to have to be kind of in relationship to each other, right? So, the largest or larger than and smaller than, but you can't, you're not going to be able to say the volume is a half a liter. That's ridiculous. What are you going to think about when you're coming up with your hypotheses? How are you, what's the core thought that's going to drive your hypothesis or your conjecturing?
Like what is actually being consumed?
Right. Well, so we know what's being consumed. So how is that going to affect these dependent variables?
Are you asking us for like to take a shot at a specific mechanism by or just say in general,
In general,
does this
I
okay,
want you
so
to I want you to formulate your hypotheses based on just the big picture of what the kidneys are doing, what the urinary system is doing. I wouldn't worry about any of the real details of the Lupa Henley or something. I want you to think more big picture about what's going on in the urinary system and how that would motivate your hypotheses.
so then like an example would be I expect somebody who drinks 800 milliliters of water. I expect that drinking that much would increase volume, decrease specific gravity, decrease salt concentration.
Right. And then
That
you
would
always
be
have
one
to have the
thing.
why part as well, because
um, because it increases the amount of fluid flow in the body, which is going to, um, I mean, you said not to get too in depth on the mechanisms, right?
right. But
But
I
you
would say,
just,
but think
yeah,
more about like the functions when we've got like the very our introduction to the urinary system. We
yeah,
talked
okay.
about what is the urinary system doing.
It's controlling overall levels of fluid in the body, overall water content in the body. So if
Exactly.
we, okay,
Yeah, so that's it. No, so if you're taking in a big excess of water in order to maintain your body's fluid balance and osmolarity, what are you going to have to do?
P.
Yeah, release more of the water. So and that shows up as pee. The kidneys removing water from your body is going to manifest as an increased volume of urine. So I want you all to think about it in terms of just maintaining homeostasis, right? If you are taking in a lot of water, you're probably going to have to let more water go to maintain your water homeostasis. If you're not taking in much water at all, the chances are you're going to have to conserve your water to maintain homeostasis, which means what about your volume of urine? Going to be decreased. You're not going to pee much at all. And what would you expect kind of the density if you don't have much volume at all? What do you expect the specific gravity going to be like?
It's going to increase.
Right. It's like because your body still has to get rid of stuff. It still's got to get rid of urea and this and that. So if you have less volume to dissolve stuff in to actually get it out of your body, you're going to expect kind of a higher concentration, a higher density of stuff in there. Right. What about salt? People who don't take salt tablets versus do take salt tablets. How could that affect the composition of your urine?
If you take in salt, the water in your body is going to be drawn towards it. So it's going to hold on to water. So you're probably not going to produce as much urine. No.
So there is something when your body has more salt, it does hold on to water more, but think of our independent or dependent variables. There's volume, there's specific gravity, and there's sodium chloride concentration. So these things are kind of independent in some ways, right? Because we have one person or one group that has a bunch of water and salt and a group that has very little water and salt. They obviously are going to have pretty different outcomes in probably the volume. If from what John was saying, no matter how much salt you've been taking, if you just drank a huge glass of water versus tiny few drops of water, this group is probably going to have more volume. regardless of whether there's salt tablets. So what else is going to be different as well?
The specific gravity.
And
The sodium chloride concentration.
right, remember, because that's the other big thing we were talking about was electrolyte balance. Right, if your kidneys, when your kidneys are adjusting the filtrate, doing the reabsorption and secretion, they are trying to make sure that your levels of sodium chloride and other electrolytes are in homeostatic balance. So how would you expect group 1 and 2 to be different from each other in terms of what you're going to see?
Group 2 will have more sodium chloride in the urine.
Yeah, you're probably going to have more sodium chloride because they're going to have to release sodium chloride in order to maintain their electrolyte balance. And then going back to the previous point, probably the volume is going to probably be a little smaller as we go along because the salt in their body while they're in this process is going to probably reduce quite how much water is leaving, because salt does keep the water in the body because it's that osmotic thing. So does this make sense how to think about the hypotheses? I want you to kind of think big picture about the kidneys doing water balance and doing electrolyte salt balance. And then think about what did these people put in their body? What are the kidneys probably going to do to maintain homeostasis over the next couple of hours? And how is that going to be reflected in these properties of the urine that you measure? So is that any questions about that?
I do have a question. We're not coming into lab on Tuesday, correct? So we're gonna.
It's gonna be all on video. You'll get to watch your instructors pee. No, actually, that will have happened off screen, but you'll get to
Great.
see your instructors play with their pee. Yeah, no, I mean, again, I'm trying to figure out what labs to do and how often to come in. At some level, and plus I wasn't seeing how well it's working out and realizing that we're not getting shut down. I probably would have scheduled even more in-person labs, but we were terrified that we were going to do all this planning and then it was just going to get rug ripped out from under us again, in which case it's like, so, next semester it'll all be in person again. All right. So any questions?
I have one more quick question. Do we have to have 4 separate, because parts of the hypothesis?
I would say probably 12, because for each group, there's 3 variables. Right, so for group 1, I expect the volume to be blah because, and the salt to be because, and the specific gravity be because. For group 2, I expect blah to be because, and again, like I said, it's going to have to be relative, right? Don't say like larger or say larger than what, because larger it has to be with, a lot of times people say I'll expect a larger volume. It's like, unless you tell me larger than what, it's a meaningless word. So I expect, the volume to be larger than groups 2 and 3 or something, but, so something like that.
Along those lines, comparing to like a baseline, how are you guys handling caffeine? Do you, like, say it was me coming in and I was a subject and I was going to drink caffeine, would I drink coffee in the morning and certainly try and drink some before this class.
It's a source of variability.
For sure.
Right. So we
do you guys ask that, or are you guys going to like wait until it's lab time to actually drink your normal morning coffee, for example, or will you
we use
it on
like,
top?
No, we use Vivarin or no dose, just those like 200 mg tablets of caffeine. Which can be, I mean, it can be hilarious. The people who aren't used to caffeine take them and they're just like, it's as good as any kind of, whoa, this is amazing. Other people, it's like, oh, whatever. But yeah, 200 megs is like, yeah, it's like one or two really strong cups of coffee. It's people, you'll feel it, and people who aren't used to caffeine really feel it. And so we get kind of high levels of caffeine in their body while we're doing the data collection. So that's how we do it. But that is still a huge source of variability around whether they're habituated to it because they just drink coffee every day, or they're not used to drinking coffee or caffeine, I should say.
So for the lab though, you would administer that on top of or just consider it. What I'm getting at is like, is there some way that you can control whether that is the first bit of caffeine that person, let's say they're a regular coffee drinker?
Yeah, no, that's just a source of variability.
Okay.
I mean, if we were doing this in a more controlled setting and it was a fancier like experiment that we were going to like really write up and control really, then we would, but not for this class. For this class, we just have like in front of them, it's like, here is your salt tablets and or your caffeine tablets and your water, and go.
Cool.
Right, because the other sources of variability would be how dehydrated or hydrated someone is to start with, right? I mean, every once in a while, you get the people who drink the huge gulps of water and don't pee almost anything. And it's like, okay, we got to talk about staying hydrated here. You were really freaking dehydrated to start this lab, and that's probably not that healthy. That's actually a more likely way to end up with these kidney stones as well. If your urine is really concentrated, it's more likely that you're going to have these different solutes precipitating out and becoming kidney stones. So staying hydrated also minimizes the amount or your likelihood of getting renal calculi. But yeah, so there's lots of sources of variability here that can mess with things. The salt as well, right? Depending on some people just eat super salty things. So we don't really know the person's initial conditions. If we were really doing this better, we would control their diet and their hydration and everything right before we started, but we're not. But then you get a number of subjects, so then you still tend to get basic trends visible because all the randomness kind of cancels out and the real effects start rising up. So, all right. So any more questions about the pre-lab? It shouldn't take you that long, but I do want you to just for each one, think about those three dependent variables. And again, the why can just be, like I said, like we need to release more water to maintain water balance because we took in a bunch of water. But I want you to think about it like that. I want you to think about, oh yeah, that's what the kidneys are doing. They're like trying to maintain water balance, salt balance. And depending on what you're ingesting, they're going to have to do different things in order to accomplish that goal. And it will be reflected in what is ultimately leaving your body as urine. So, blah, blah, blah.